CERTH was founded in 2000 in Thessaloniki and is today the leading research center in Greece, ranking first in the country and 11th among research organizations in Europe in terms of competitive programs. In FUELGAE project, CERTH is represented by two groups that come from SPERI, and SPERI is one of the 5 different institutes of CERTH conducting applied research in biochemical technology and fuels, among of course other areas. One of the roles of CERTH in the project is to use its experience with different microalgae species and cultivation systems in order to develop a process that efficiently converts CO2 from industrial emissions into microalgae biomass, which then can be turned into advanced biofuels. Microalgae are photosynthetic microorganisms that, much like plants but of course more efficiently, can utilize CO2 and sunlight to produce biomass. They can capture CO2 from the air, from industrial emissions, even from simple heating systems. However, exactly as all living organisms, microalgae need specific conditions to grow well, and stress factors like too much CO2, for example, or other gases can affect them, exactly as it happens in FUELGAE project. Thus, we are presently working to select and improve microalgae strains that can handle these tough conditions that are applied in the project case studies. These case studies are bioethanol and steel plants. So after selecting the right microalgae, the project will focus on improving them via different tools and methodologies. As we speak, our partner VTT in Finland is using adaptive laboratory evolution to create stronger variants of the selected species. Then CERTH will perform design of experiments for these species in order to optimize the operational conditions. These activities, including the process scale-up, will follow the specifications of the 2 project case studies. Eventually, we will focus on adjusting the species for specific challenges, like the high CO2 levels from the biorefinery and the high temperature of emissions from the steel plant. During the project, the proposed technology will be validated up to TRL 5, meaning that there is still considerable work to be done following the hopefully successful completion of FUELGAE. The most important application of these improved microalgae species will be their ability to directly capture CO2 from industrial emissions, helping reduce greenhouse gas emissions. For this reason, the proposed technology will include also a compact, modular and scalable photobioreactor. The flexibility of the selected microalgae to adapt to different industrial conditions, once of course fully optimized, will allow this technology to be used in a range of industrial conditions and industries with different emission profiles. Next, CERTH will further optimize the cultivation conditions for the selected microalgae strains. We want to make sure that they can efficiently capture CO2 and produce biofuels under industrial conditions. We will also focus on scaling up from the lab scale to the pilot scale photobioreactor. This will allow us to evaluate how well the optimized strains will perform in larger volumes under real-world conditions. By the end of the project, we aim to show the full potential of these strains to reduce industrial emissions and support sustainable biofuel production.